So now we're going to move on into section 6, which is the historical foundations of microbiology. And so the learning outcomes would be to make a timeline of the development of microbiology from the 1600s to today. And I'm not going to ask you to do that for the test, but you should know kind of relatively how early were these developments, and maybe not specific years, but just know kind of a general timeline or general idea of when these developments occurred and who were involved, who were the people involved. List some recent microbial discoveries of great impact, and then explain what is important about the scientific method. So some early ideas about food were that, the food spoils, or about disease transmission. We look at foods and, it was spoiled, so they didn't eat it. It was inedible, and so, or it caused illness, and so we knew that food could get contaminated. The Black Plague and smallpox caused by some kind of transmissible matter. They blamed it on the rats. It wasn't actually the rats. It was a bacteria on a flea on a rat that caused the plague. And smallpox was a virus that nobody could really see. So they didn't understand how it was transmitted at the time. And there was this belief in spontaneous generation, which is also called the theory of abiogenesis, which is abiogenesis. A means the absence of, bio, life, genesis, the creation. So biogenesis. So the absence or no life, creation. So life created from nothing. So in the 1800s, they finally put the nail in the coffin of this, that, life cannot come from nothing, that it has to come from something. So Pasteur was the one that pretty much disproved this. There were some other people. He had this swan neck experiment. So this swan neck experiment basically said that as long as it's intact, microbes can't get in here, so it will stay sterile for the rest of its days, until you break the neck or something happens where the liquid comes into contact with this area right here. The broth is going to remain sterile. So it's open to the air, so it can get air, but the microbes can't get past that little crook in the neck. So there were some other people. There was a guy named Reddy, who was the meat and maggots guy, who said that, he had some meat and he put it into 2 little containers, put the little meat in there, and we'll just say that that looks like meat. And he had one that was covered and one that was not covered, and this remained. So this had no maggots because it was covered, and this one had maggots because it was uncovered. And so he said that life can't come from nothing, that the flies have to be able to touch the meat to be able to lay their eggs, and the maggots to be able to develop from that. And then there were some others that Schleiden and Schwann had this kind of elaborate setup where they heated the air going in, and they said, well, no, that really doesn't do it. So when Pasteur did this without heating the air coming in, and showed that it would stay sterile as long as the neck was intact, that was kind of the final nail in the coffin. Then, to be able to really study the microorganisms, we had to be able to see them. So the first observations of microbes were actually attributed to Robert Hooke. So he was what I kind of say that he coined the term cell because he saw cells in so he coined the term cell because he was looking at cork and other types of organisms, but mostly cork, and he saw these little tiny places where the cells were no longer there in the cork, and figured they look like prison cells or cells, the rooms that monks live in monastery, and he said that these look like cells. So he coined the term. He was the one that gave us the term cell. But he was also the first person to make observations of microbes in the 1600s. And then Antony van Leeuwenhoek was around the same time, and he examined fabrics with this crude microscope that he made right here. So there's a little hole right there, and then there's a specimen holder right here, and he could move that up and down and look through the hole with the lens in it. And so the amazing thing about this is that the single lens, he had one lens, and it was able to magnify 500 times is the estimate that they have for this. But in actuality, you know, the microscopes that we have, the most that we see is like 100 times, maybe 1500, or 1500 if you have like a 15x ocular lens and a 100x, or you could get 2500 if you have a 25x, you know, ocular lens and a 100x. But, you know, one single lens, not a combination of lenses, giving us a 500x magnification is pretty amazing, especially considering he was back in the 1600s as well. So Robert Hooke and Antony van Leeuwenhoek were these the people that were really instrumental in developing the microscope. And then we've refined the microscope. The lenses are now able to go from edge to edge, and they're called plan achromatic, so they don't have these chromatic aberrations because of the edges of the microscope lens. The picture is edge to edge, clear, and in focus. And they have built-in light sources. When I was in high school, we used little mirrors to get the light from the room into the microscope, or we had to be out next to the windows to be able to see. Yeah, it's not like that anymore. And we have much more extensive microscopes, so we'll talk about that when we get into the microscope lab next time. So the century of biology, we've seen restriction enzymes, which those are enzymes that can cut DNA, and this led to recombinant DNA techniques. We were able to add a piece of DNA from one organism and put it with another organism, as long as they use the same genetic scissors. And then we developed the PCR technique, which is basically like a DNA copy machine. So it can make many, many copies of DNA. We learned about in the 2000s about the importance of small RNAs like microRNAs, and silencing or small interfering RNAs, and how they can shut off certain genes and stop genes from being expressed. And then in the 2010s, we learned about how important the microbiome is and how involved that is in cause of disease. And so this has been the latest kind of new thing. So I would say probably the next coming thing is going to be probably CRISPR, which is a gene editing tool. Which uses basically the DNA of, or not DNA, but enzymes from a bacteria cell that the bacteria use as an immune system to prevent viral infections from coming back if they get infected by a virus. Again, the CRISPR enzymes will cut up the DNA of the or RNA, any nucleic acid of the virus. And so we are using it as a gene editing tool, like a find and replace thing in our genome. And that has been going on since 2012. So that's probably the newest thing. And it's related to microorganisms because it comes from microorganisms. So the establishment of the scientific method, that was a big finding and was the result of then newer findings and the ages, the golden ages of microbiology would not have been possible if they had not established the scientific method. But the general approach that scientists use to explain natural phenomenon, that is a scientific method. And a tentative explanation, a potential explanation of, to account for what has been seen or measured, something natural occurrence in the natural phenomenon. So a tentative explanation for some natural phenomenon is a hypothesis. So just a little bit about deductive and inductive reasoning here. So a deductive reasoning would be when you have general principles that then explain specific observations. So you have one general principle up here, and it can describe several different observations. And inductive reasoning, you have several observations that then lead up to one general principle. So specific to general, general to specific. And I don't know that you have to know if it's an upward arrow or a downward arrow, but that's just the way I look at it. So you have one general principle that explains lots of different observations, and where inductive reasoning you have lots of observations and they lead to a general principle. When we do hypothesis testing, we usually have lots of different results, and that leads us to a general principle, but it could be the other way around as well. So you make observations through experimentation, and then that leads to a general principle that is like your theory or a law if it's been supported by lots of different people and lots of different observations. So here's another way to look at that. When you have deductive reasoning, you look at your computer and you have something that looks very suspicious, and you know that opening attachments from unknown senders that look suspicious can introduce a virus to your computer. So you know this to be true. That's a general principle. And so you have a specific action based on that, which is to not open your attachment. With inductive reasoning, you do this over and over again. You open up documents and you click on them, and you get viruses on your computer. And so then you come up with this idea that opening up unknown attachments can damage your computer because you've done it before, you've experienced this, you've observed this many different times, and that leads you to this general conclusion that you should not open up these attachments from unknown persons. So that is the way that we talk about deductive and inductive reasoning. So I'm really wanting you to know, though, the difference between a hypothesis and a theory. So again, a hypothesis is a proposed explanation for natural phenomenon, and it's based on previous observations or experiments. You make a prediction that can be either shown to be correct or incorrect, so it's not always correct. You either support or reject a hypothesis, but you don't really prove it. Like maple leaves drop their leaves in autumn because of shortened hours of sunlight. You've noticed that the leaves drop their, the trees drop their leaves in autumn, and you've also noticed that, oh, at that same time, the hours shorten, sunlight hours shorten. And so therefore, that must be the cause. So that's a testable theory or a testable hypothesis. So a theory is a broad explanation for some aspect of the natural world that is substantiated by a large body of evidence, and it allows us to make predictions, and that generally those predictions are true. So consistent with and so the two attributes of a theory are that they are consistent with a large amount of data, and you're able to make correct predictions. Where with a hypothesis, it's one or two observations. So previous experiments are small, amounts of observations, and you can make a prediction, but it may be incorrect. Where when you make a prediction from a theory, it's usually correct. It allows you to make correct predictions. So I do want you to be able to differentiate between a hypothesis and a theory. In biology, you need to be very curious. There's no really rigid set of steps. There are generally two approaches to the science, discovery-based and hypothesis testing. So discovery-based, you gather information and, like, for example, testing drugs to see which one is effective against a particular disease, sequencing genomes to see what the sequence is, or what the proteomes, what are the proteins in this organism. And that often leads to hypothesis testing. And hypothesis testing, you have observations that lead to a hypothesis that you test, and then you gather the data and you analyze the data, and then you have conclusions. So those are the 5 stages generally of hypothesis testing. And a lot of times we would call this the scientific method, but there are, again, a lot of medical discoveries or medical papers. If you look at them, they don't say the hypothesis for this is this. It's usually a lot of this discovery-based science that's going on. So science and its hypotheses and theories must progress with technology. We know that as the instrumentation advances, more detailed view of things, and we can, re-examine old theories and alter them and propose new ones. And so this is continually happening. It's, not a set thing for the most part. We have to be flexible enough to realize that as we get new data and we have new instrumentation coming in, that we may have to change what we believe about things. As you could see, like with the idea of Helicobacter pylori, we used to think that gastric ulcers were caused by stress. Now we know, because we've been able to isolate and sequence the genome of bacteria that are in the stomach, we know that it is, and we've been able to prove and show through testing that, or not prove, but support our hypotheses, that it's actually Helicobacter pylori, the organism that causes gastric ulcers. So you have to be curious, open-minded, have some skepticism and creativity and cooperation, and be able to revise and be ready to revise your views of natural processes as new discoveries are made. So in medical microbiology, early experiments showed that microbes are everywhere, and the air and dust are full of them, and that the entire surface of the earth and planet is all covered and inhabited by them. John Tyndall and Ferdinand Cohn had something to do with the discovery of spores. So Tyndall found that microbes in the dust and the air have, some of them have a high heat resistance. And Ferdinand Cohn discovered bacterial endospores. So there are some bacteria that maybe don't produce endospores, but they have a really high heat resistance, so they are not always killed by heat, or the heat has to be hotter than what we were heating them at. Like boiling water doesn't always kill everything. If they have endospores or they have a high heat resistance, they could tolerate that and they could still be viable. But endospores are these, in an organism that produces endospores, they become sporulated. They produce a spore inside the cell, and then the outside of the cell dies eventually, and you're left with a spore that can then germinate and produce a new spore. But it's not a method of reproduction like pollen or other spores for plants and fungi. This is an endospore. It forms one spore in one cell, and then one spore can produce one cell. And so it's just a method of survival. But Ferdinand Cohn discovered these and said that something that is sterile is completely free of all life forms, including spores and virus particles. And so then we had to figure out how to do things to sterilize instruments, to sterilize liquids, and Tyndallization actually did develop from Tyndall's idea about how we can kill most of the pathogens, not all of them, but over a period of a couple of days. And then pasteurization developed from Pasteur's looking at fermentation, and he figured out a way to prevent wine from becoming spoiled, becoming acidic. Instead of becoming acidic, which is caused by bacteria, he wanted to save the yeast, but not allow bacteria to grow in the wine. So the development of aseptic technique, Louis Pasteur came up with his pasteurization process, where you have short bursts of heat that kills the pathogens, although not all the microbes, so it retains the flavor of like liquids such as alcohol, wine in particular, because he was from France and it was a winemaking country. And milk would be another one, so that killed most of the pathogens, but not all, or most of the microbes, just mainly the pathogens that they were more concerned about, but not all the microbes. And then he also developed the germ theory of disease, which said that one germ results in one disease, although he did not prove it. And then Robert Koch came along and proved his germ theory of disease by developing these postulates. He was able to identify the causative agent of the diseases. And this kind of opened up a whole new kind of golden age of microbiology, because everybody was in a race to see who could develop and find the next disease causing microorganism. And then other development of aseptic techniques involved Oliver Wendell Holmes and Ignaz Semmelweis. And essentially, they both figured out the same thing at the same time, and they observed the mothers who gave birth at home experienced fewer infections than mothers who gave birth in a hospital. And then Ignaz Semmelweis observed that physicians who were working in autopsy went directly to the maternity ward and without washing their hands did a pelvic exam, and so then the women became infected. So this is, both of them kind of figured out we need to wash our hands before we deal with pregnant women and other patients, and so they enforced hand washing in their physicians as a result. Not a very popular idea when they came up with this, because who wants to be told that you're the cause of people, especially mothers, getting infected? So then Joseph Lister came along, and Joseph Lister gave us aseptic technique in surgery. So you can see he was the first one to utilize hand washing and misting in operating rooms with antiseptic chemicals, not just soap and water, and techniques that we now use today. They were the foundation of modern microbial control that's still in use today. So spontaneous generation was disproven by Blank with his experiment using swan-necked flasks. Polymerase chain reaction was invented in what decade? When a hypothesis is supported by a growing body of data and survives rigorous scrutiny, it is known as a Blank. And question 4, aseptic techniques were developed by Blank.